So this is a topic, we're talking about regulatory trends with E&L, what we hear from the FDA. This is one of these like hot button issues in med device right now. There's really a lot going on with medical device extractables and leachables. A lot has changed over the last 2.5 years. We see a lot of stuff going on even in the last 6 months. It's really open-ended, this topic. I hope in this format, I know it's a little bit hard to like have dialogue and ask questions or whatever, but certainly there's a lot that we could talk about. I'd love to, please feel free to interrupt me or whatever as we go along. If you do have any questions or something special that you want to talk about. Yeah, because it's one of these things, we interface with the FDA a lot at Nielsen Labs. In my role as the manager of our toxicology consultants, we do tox assessments for many labs everywhere. And so we see lots of chemistry reports. We interface with the FDA on a weekly basis for our own chemistry studies and others. So we have a lot of exposure here. There's a lot that we could talk about. Okay, but before I get into that, just a very brief personal introduction. This is the same slide that I showed over there for the center stage. My background is in academics. I'm a nerd through and through. If you want to talk about photonic materials, you'll get me really excited and we can have a nice chat. That's really has been my passion before I got into the medical device field. So what I love about med device, I've been doing this for about 6 years, is that I get to apply this like skill set and knowledge with the physics and materials to something that actually to me matters. Making devices safer for patients. I get to help manufacturers get their devices to market sooner. That to me is a pretty meaningful thing. So I've been doing med device for 6 years. I studied toxicology in the meantime. I'm now a board certified toxicologist, so that's I do quite a lot of that. My one true claim to fame is that I helped create a flavor of Mountain Dew. That's Mountain Dew Whiteout. It's a delicious flavor that is sadly tapering off the market. I still see it every once in a while, but it's getting hard to find. So if you see a Mountain Dew Whiteout, pick it up, enjoy the refreshing smoothness while you can, because it's not going to be around for very much longer. Part of the reason why I do this, so first of all, I told that I got the nickname Dr. Dew because of this. It was given to me by some of my students back when I was teaching at the university level. I was like so enamored by it. I've continued to use it. It's also functional as well, because my name is Matt, and there's like a zillion Matts, really many Matts. So Dr. Dew also is kind of nice to distinguish which Matt that you're talking about. So to lead into this, before we talk about extractables and leachables and what we're hearing from the FDA, I think we really have to put it into context with a little bit of toxicology. That the whole reason why we do medical device extractables is so that we can leverage that data using toxicology to make certain claims about safety, right? And so it's actually the demands of toxicology and what the toxicologists at the FDA expect that sets the criteria for what's acceptable for an extractable study. And that's important to remember. So we'll start with that. I'll get into some pieces of FDA feedback that I copied and pasted into here, but this is like, 3 or 4 slides worth out of like 1,000 possible slides. So if there's any like special interest with FDA feedback that you might have, or you're curious about, what's the FDA thinking about whatever, please feel free to just stop me and ask, because I'd rather talk about what your concerns are than like what I more or less randomly selected. So here's kind of the background here. We have instances like this, what's shown on the screen. So, we have case studies in the literature for medicine where people are showing up at hospitals sick. They've been poisoned by their medical device. And again, this is like one example out of like, many that you could choose. You have the whole thing. It's not extractables, but everybody remembers Sulzer Orthopedics and the issue with those residuals that they had on their orthopedic implants. This is a case with cobalt poisoning. Of course, it's a metal on metal device, but the idea that we can be poisoned by our medical devices, this is acute to the FDA. They're very aware of this concern. They want to make sure that we know that our medical devices aren't going to poison people. And I think it's a fair question to ask. It makes sense that they're concerned about it. Now that we know that that's a concern, actually proving that and bearing that out is where this challenge of extractables and tox assessment for extractables comes from. In order to try to explain that, I think we have to like I said, do just a teeny bit of toxicology. I talked a little bit earlier about biological evaluation plans. If depending on the type of device that you have, if you have something that's permanently contacting to tissue, bone, or dentine, for example, then you have a suite of biological risks that have been identified and that the FDA agrees are risks. And so, for example, the ones listed there. So I also put hemocompatibility on there. That's in case it's in contact with circulating blood, direct or indirect. And of those risks, some of those risks are specifically systemic toxicities. So when I say systemic toxicity, this means related to compounds that are dissolvable in the body that can get distributed throughout the entire body, either by the vascular system or the lymphatic system. So all of those that are in green with the arrows, those are those types of risks related to being distributed through the body. And the FDA has explicitly said, the ones with the arrows, that they will take extractables testing and a toxicological risk assessment as a means of addressing those biological risks. The other ones, they explicitly say no. So at least in CDRH. So on the medical device side, sensitization, you can't use chemistry to address. On the pharma side, you absolutely can, right? So we have great information on sensitizers. So this is really med device specific, what the FDA will accept. So if we're going to do this, like be in the business of saying, okay, my device, this beautiful pacemaker, doesn't cause cancer. How do we actually do that? And this brings me to this one slide intro to toxicology. It's really more like a slide and a half. So toxicology is all about one thing. How much of a good thing is too much? You hear toxicologists say it over and over again, the dose makes the poison. And that is absolutely correct. Everything is a poison at some dose, and at a low enough dose, things aren't a poison, right? And so I give this as an example. This is like very personal to me. As you know, I drink a lot of Mountain Dew. This is probably number like 4 for this afternoon, And so I know I've experimented on myself for all of you, like how much caffeine can I tolerate as a service, really. And so I know how this looks. So if I have no caffeine, this is actually causing a negative effect, right? So I'll have headaches and be crabby. And I did go off caffeine for a couple of weeks once, and my coworkers literally begged me to get back on. I was just so irritable. So, and you can have this. There's like homeostasis with things like vitamins, right? So if you have no vitamin, that's a problem. If you have too much vitamin, then that's also a problem, right? So it's the same for me. There's this new normal, that's probably where I'm at like right now, like a minima of adverse events. But if you continue going up, then more and more things start to happen that could be a problem, right? So, you reach a point with caffeine where you can have arrhythmia and tremors. So I have been to the the tremory stage one time when our very cheap event organizer bought like 100 energy drinks, and it was use them or lose them. So he kept on trying to get me to like consume them so they wouldn't go to waste. He's standing right over there, right over there. But so he kept, and in the end, it was like 6 Red Bulls. And then I was like, okay, whoa, 6 is like 2 Red Bulls too many. So you have tremors, hallucinations, and finally you reach this point where there's like 50% chance of something really bad happening, for example, death. And then you reach a plateau where it's certain that whatever bad is going to happen happens, and an increased dose can't do anything more than what it did. Everybody died. And then for caffeine, that's about 150 milligrams per kilogram. I think I calculated one time, it's like 110 Red Bulls, something like that. So that's a lot. For in general, for foreign chemicals, things that might be a contaminant on a medical device, the curve looks more like this, where if you aren't exposed, nothing bad has happened, that's great, it's nothing. If you have a very small amount, there's nothing measurable that can happen. And they do these studies in animals where they give increasingly higher doses until they start to see an effect. And the highest dose where nothing bad had happened, that's the no observed adverse effect level, the NOEL. And then they really should measure at least one data point beyond that so they can know that their test system worked, and that's the lowest observed adverse effect level. So they're trying to say, okay, how much of this foreign chemical can an animal take before you start to see something negative happen? And something negative could be like weight gain or a change in organ weights, things like that. And then you can continue on. So like in acute studies, you can have this dose where something bad happened 50% of the time. The most common one is the lethal dose 50. That's enough that 50% of an animal population had died. Okay, so this type of data, what I show on the screen, is like embodied in the literature and databases. If you go to ECHA's database, they have a really, really great huge body of this information that anybody can access for free, right? The goal that with toxicology and the reason why we're doing this is we say, okay, if we can measure these chemicals, we can look up the chemicals in the database, find these S curves as they're called, and we just want to be sure that however much of, let's say DHP, however much of DEHP that we find, it's like below or sufficiently below that NOEL, so that we can say, okay, this much of that compound won't cause anything bad to happen. So recognizing that that's the goal, that sets up this big chemistry problem. It turns out to be like a really gnarly, complicated chemistry problem. So we want to be able to find anything that might leave the device and also measure it with enough accuracy and low enough that we can compare it to all of these curves. Yeah, it's tricky. And so the way that we do that, I guess, we have this, I want to explain, I guess, the terminology, extractables and leachables just a little bit. So the notion is, if this device is in the body, so this is a real device, it's a 3D printed Spock hand. It's designed to be implanted in the body next to the heart so you can keep the memory of Leonard Nimoy with you always. If you are the Trekkie that gets this implanted, after it's implanted, compounds will slowly leave, and those are the ones that we really want to measure. It's not really a surprise that inside your body, it's like a complicated mixture of chemicals that are around a medical device. So it's not like there's water, right? Because we're mostly water, but there's also like fats and like lipids and all these other things that could dissolve compounds. And so that's what we mean when we say extractables. These are anything that could theoretically leave the device using really aggressive conditions during use. When we say leachables, that's what should actually leave during real clinical use. Now, we have to be a little bit careful there because in a pharma system, let's say like a single-use system for biopharmaceutical processing, this is something that we can actually measure, right? Because we can simulate exactly what those things are exposed to, or a pharma container, like a vial for an injectable. But in the body, we can't, right? So we can't put a device in the body and then actually measure these chemicals out of somebody's blood or fatty tissue, because the blood and fatty tissue ruins the analysis. So in pharma, this is possible exactly. In medical device, it's almost always impossible. There are some cases where you can work it out. Really what we mean when we say leachable, we mean more like simulated use extractable, where we're going to try to mimic clinical use as close as we can, recognizing that if it's a brain implant, we're not going to get an actual animal brain and put the thing in there, right? Okay, so that's extractables and leachables. As I said, we need to extract as many compounds as possible because it's difficult to predict what might come out. Analytically, we cast a really, really wide net looking for everything that could come off of the device. From a chemistry perspective, that's a really challenging problem. So we're not looking for just a list of plasticizers or a short list of metals. We need an open scan type methodology. And this is something that the FDA has been very clear on, and they continue to sort of refine and expand what they mean by an open scan. So for example, I don't think I have this exact feedback coming up, but they'll say, okay, you did LCMS with ESI ionization, but we're worried you could have missed some things doing ESI ionization. So we want you to do APCI and ESI, regardless of Dr. Jorgensen saying that doesn't make sense, right? So they look for ways to expand that scan. That's the open-ended part of this looking process. The other thing is this sensitivity, right? So this is where things get even harder. So what we are worried about, so chemicals that we detect and we can estimate their concentration, we know what those chemicals are, we can look up those S curves from the literature and compare that amount to what was in the literature. But then, a regulator may say, well, how do you know that you looked low enough that you didn't accidentally miss something that could be potentially concerning? And we would say, well, how do we know how low to look if we don't identify it first? It's kind of like a chicken and egg thing. And so we have this idea called the threshold of toxicological concern. It's basically, if you imagine the most toxic thing ever, and in this case, it's like compounds that are used as positive controls in carcinogenicity animal studies, so compounds known to cause cancer every time. So you take these very, very nasty chemicals and you know how much of those would be needed to cause a problem, and then you divide that by a huge factor. You say, okay, if anything is present that low or below, we know it's not going to be a problem. Even if we don't identify it, even if it's an unknown, if it's below this threshold of toxicological concern, fine. So we were doing that. We were doing that for a few years until about 3 years ago, when the FDA was like, what about the fact that when you screen, it's more like an estimated concentration? And so actually, you're kind of like, there's a little bit of wiggle room when you estimate how much of that chemical is there, and you could be off. So maybe it's reported at 1 microgram per device, but since we know that in screening methods this estimation can be a little bit off, it might actually be at 3 micrograms per device. And then it would be above the TTC. So now we take the TTC, this threshold of toxicological concern, and divide it by an uncertainty factor that accounts for the fact that when we screen and we don't have an authentic standard for everything that might randomly come from a device, that whatever we see, even considering this uncertainty, wouldn't be above the TTC. So this thing about the AET is really, really important to the FDA. I've seen devices, good devices, life-saving devices with excellent data be completely denied clearance by the FDA on this basis just alone. And it's not like these guys were really far off. It was okay, it should be 0.75, but instead it was 1. So this is something to take seriously. You don't want to get there at the end, after you've done all this biocompatibility work, you've done a chemistry study that took a really, really long time, and then a toxicological risk assessment to have the FDA say, no, it's not sensitive enough. Because the only recourse there is to redo it, right? You got to do the chemistry testing again, but more sensitive. So this is an important idea. I've been kind of going really fast. I have more to talk about, but since the AET is so important, is there any question about the AET and its role in extractables and leachables and how serious the FDA feels about it? Yeah. That's an excellent question. So the question was, is that uncertainty factor the same across the board? Or does it matter on the lab or case by case? And the answer to that is that, again, this is something that has evolved, and it's important. So 2 years ago, when we started to do the AET, there's some papers. Dennis and Janke and a couple of other ones have measured these uncertainty factors in exemplary cases. And there they had something like an uncertainty factor of 4 for GCMS and an uncertainty factor of 10 for LCMS. So people were using that, but the FDA threw I don't know if you want to call it a red flag or a yellow flag. I'm going to say a red flag. Said, no. Like, your lab isn't the same as Dennis Jenke's lab when he made that publication, so you need to figure this out for yourself. And so it's on a lab by lab basis. This number needs to be computed and supported. And it's one of those things where it's not like you can compute it just any way according to the standard. You have to compute it in the FDA's way, right? So you need to have that data and then make sure that you compute it in the FDA's way, not necessarily any way that the standard supports. So the way that it is operating today is that every lab that we deal with has computed this for themselves on their own instruments in their own space and are defending that uncertainty factor is good enough. It's a really good question. Any other questions about AET stuff?
Follow on to that. So nobody's just throwing a high uncertainty factor in just to, nobody ever did it higher than 10, so I'm going to use 20. Set my AET based on that. Go home early, or it has to be?
So for reals, we were in that exact spot where we had some evidence to suggest that our uncertainty factor was not as high as 10. But it was like, okay, we're going to do all of this work that the FDA is asking to prove that it's not as high as 10, or should we just use 10? So we used 10 knowing that it was worst case. The FDA said no. They're like, we love that you say that it's worst case, but no, we want to know the actual number, which was painful, but now we have it. Any other AET questions? Okay. We keep on going then. I want to talk then, pivoting a little bit about exposure, because this is now a little bit forward looking, what we think is going to change in the future. It's all about exposure, right? So right now, the expectation today from the FDA is that if you're going to do medical device extractables, it should be exhaustive. And exhaustive means that you extract the device over and over iteratively until greater than 10%, greater than 90% of everything that could come off has come off. So you do like 24-hour extractions, you'll measure it by NVR. NVR is like my non-volatile residuals. That's like my least favorite method. I can like right now rattle off like 10 reasons, or maybe not 10, 5 reasons why it's just so dumb. But the FDA really, really wants NVR. Even if you can do it a better way, they still want NVR. So iterative extractions, measured by NVR until it's less than 10% of whatever the initial or maximum was. And then you extract for that long, or you do multiple extractions that add up to that long. And that entire amount that came from the device is supposed to be assumed to be the chronic lifetime daily exposure. This is really, really conservative. And to me, it doesn't make a whole lot of sense. It's like, well, why belabor us to prove that all of this came off if now we're going to ignore that and say, wait, it comes off every day forever. But that is the expectation. So I've seen it from the FDA over and over. Now, if you got to be at least attempting this. Now, if you do this and the amounts are too high, you can say, okay, we know that's crazy. Let's do something a little bit more realistic, like a simulated use, or look at the kinetics. But I've seen cases where we've led with kinetics, not we, person we were working with, led with kinetics, and the FDA said no. So they did 24-hour iterative extractions. We showed it. They said, yeah, but what about exhaustive? So that's what this is really looking like. So exhaustive is the way they expect it. We used to do more regularly exaggerated. That means 50 degrees C for 72 hours. That's one of the criteria in the standard. It should make sense. The FDA really isn't taking this instead of exhaustive anymore. That's what this looks like, where you measure it over these 3 days and you assume that exaggerated amount is the chronic lifetime daily exposure. The way that I think that it's going to go is closer to what I showed there, the simulated use. This is the way that makes the most sense to me by like a long shot, is if we know that the dose makes the poison, and when we say dose, we mean the amount of compound per day in total. That's probably ought to be closer to what we're measuring, right? The amount per day. And so that's what a simulated use where you do multiple iterations would look like. So we see this, in the pipeline. If we look at the type of talk in the 10993-17 committee, this is the standard for toxicological risk assessment. We want to put this kinetic information in there so that we're really thinking about the exposure rather than this exhaustive amount being the chronic lifetime daily exposure. So that's something that we think is coming. Is there any questions about exposures and the way that the FDA wants us to assume, what we measure, how it relates to an exposure? Okay, let's kind of marinate on it a little bit. I can see some people thinking about it. Okay, so here's some a more or less random grab bag of FDA feedback on E&L studies that I pulled quickly. This one is interesting, right? So it says it's not clear how you calibrated your GC instrumentation. The FDA recommends a minimum of 5-point calibration curves for semi-quantitation. You should provide calibration data that demonstrates suitability of the calibration curve across a concentration range of extractables. You can see there they talk about linearity, goodness of fit, all of this stuff. So this is something that we've seen more and more. It's really a lot of work to supply this information to the FDA. It takes up many pages, but this is something that if you're going down this path, whatever lab you use, they should be ready to give this to the FDA if the FDA asks, or they should just be providing it as part of, as an appendix to their chemistry reports. The way that this looks most often now is that when we talk about 5-point calibration curves, this corresponds to surrogates or internal standards that are part of each of those analyses. And for GCMS, it's usually a minimum of 3 surrogates or internal standards. It's not really a true internal standard. I'm going to use surrogate. Minimum of 3 surrogates that have this criteria met, where there's a calibration curve. The calibration curve has at least 5 points, and the range of that curve goes down low enough to measure the AET. And those surrogates should have a range of properties so that you could argue that, we have a plasticizer, we have this thing, and that means that nothing's really falling off the charts here. And so for each method, there should be a minimum of those surrogates where this criteria is met. And that's how it looks at most labs today. And if the FDA asks, they can give you this list of surrogates and this linearity and all this other stuff. So this is something that is newer and important, and it's something that the lab should be ready to provide. Okay, here's another one that I think is pretty interesting. Please note, sufficient information about potential extractables should be provided to exclude the release of some structural classes. So this gets really thorny, right? So I said, the TTC is this amount that's based off of this most toxic, crazy thing. That's all true, but in the standard, ISO 21726, where it talks about the TTC, it says, but wait, there's also some compounds that we're going to say are TTC exempt. So these are so-called cohort of concern compounds. Compounds that, under certain situations might be more toxic than the TTC or might bioaccumulate, things like that. So the FDA will say, okay, we want you to prove to us somehow that you're not having these cohort of concern compounds present. Now the way to address that isn't easy, right? So with the screening technique, we have to have some cutoff. This is how low we go. It's not impossible to have infinite sensitivity or to look infinitely low. The lower you go, the more noisy it gets until it's just all garbage. So there has to be a cutoff. If we say we can't use the TTC, then that creates a big problem. So the way that this usually looks is that if you go down the list of COC compounds, you can ask yourself, could cocaine be on my device? That's one of these COCs. It's no, it couldn't. What about aflatoxins? So that's the byproduct of the device being in contact with mold. Is our device in a moldy environment? You say no. And so you can rule out a lot of COCs until you reach some organophosphate compounds or strained heteronuclear rings. And when you get there, you can say, okay, well, let's take a look at all the other compounds found, and let's take a look at the manufacturing, processing, and the device materials, and we know that there's not this class. So you basically have to make an argument that you say, we know that those types of compounds aren't part of this. That's sufficient to argue that they couldn't be there, even though we can't really test for them that low. Okay, so to kind of like summarize some of these points, so in general, for chemistry and medical devices, it's not already like clear by my caffeine-fueled ramblings, right? The FDA has like paid attention to this a lot. It used to be, so when I started 6 years ago and people like E&L was really starting to catch on for medical device, there really wasn't a lot of scrutiny on the part of the FDA for exactly how labs were doing this. They trusted the lab that this would be done right. And then what happened is there were some comparison studies where the same device or same device materials were sent to many different labs and got very, very different results. That really freaked the FDA out. And since then, there's been progressively more scrutiny on this. So this is not something that you can do without, you just know that the FDA could pick apart any weird little detail of this study. So there's really lots of scrutiny. Now, right now, we're in an uncomfortable position because there's really a lot of scrutiny, but it's not standardized. Reviewer A isn't asking the same questions as reviewer B and so on. So what we hope soon, and we hear that, they're close, that there should be like some consensus from the FDA, written guidance, like this is what they expect from a medical device extractable study, and then everybody can just do that, and then we'll be kind of like out of this sort of like swirling storm of new feedback. I really think that there's got to be a refinement of these exposure assumptions. So that's going from exhaustive to exaggerated to like, let's take a look at the kinetics of release from medical devices. If we're going to be as sensitive as the FDA wants, we can't be that sensitive in analysis and make such an insane over-assumption on the exposure. The two are kind of like running into some problems. So I think that that's in the works. I really hope that gets into the standard. We'll see. With these compounds of concern, this is getting like, I feel like this is escalating a little bit more every day. So there's all of this attention on nitrosamines right now, right? And people are asking about nitrosamines, the TTC doesn't apply. Every time there's something about these COCs when the FDA is asking. So right now, that's what's happening. There's a lot of scrutiny there. We can kind of like cobble through that. What I think is likely to happen in the future is that there's going to be some sort of targeted panel for these ones that bother the FDA the most. We'll have like a short panel of a couple of nitrosamines, a couple of really, concerning other compound of concerns, and we'll just screen for that panel, right? So we'll take the, we'll know how sensitive it needs to be. We'll just screen for it and take that issue out of the works head on. So those are probably the three things that are like on the top level what I see going on. With that, I'll go ahead and pause or stop, and we can take any questions that you want. Before I forget, I have all sorts of useful materials up here up front, like the biocompatibility slide ruler and some other stuff. If you want a copy of this presentation, just leave me your business card. I'm happy to give you a copy. Okay, so are there any questions about what FDA is saying or thinking for medical device extractables? Yeah.
Is there any crossover from CEDAR to CDRH?
Yeah, so the question is there any crossover from CEDAR to CDRH? I really wish that there was more, because, and we start to see this with some things, like when you look at combination devices and, CEDAR and CDRH have to work together on it. And I mentioned single-use systems for biopharmaceutical processing. That's also in that realm. Right now, I don't see that much crossover. Hopefully it's happening. I would love to see it happen for things like a sensitization. That's a very crystal clear difference. CEDAR accepts chemistry for sensitization. CDRH doesn't. There's other things too where we have good arguments for a pharma audience inside the FDA, but not for CDRH.
it's exhausted, there's no more to give, so you're in equilibrium.
No, I agree. So he said that, he's talked to Cedar. They'll accept kinetic data. I absolutely agree. So we've seen that as well. It's a lot more, I think, understood and accepting there of that type of data. And I'm really hopeful that CDRH will move in that direction as well, because I think that that's a convincing argument for sure. Anything else? Yes.
When is it acceptable or to partial extractables?
Yeah, so the question was, when is it acceptable to do a partial extractables and leachables, or some sort of like subset of that? And my answer isn't going to be very satisfying. From my experience, we could do a partial with sound scientific argumentation 2 years ago. Today, even if it's like crystal clear that a partial makes sense, they still want to see evidence of the full. So what we'll do, so like, for example, imagine you have a polycaprolactone device. So IPA dissolves that material readily. So what we would have done is said, okay, we know IPA is incompatible. We're going to extract in water and hexane. Now we'll put it in IPA. We'll take a picture of it dissolved. And now we've shown, okay, you put this in IPA at 50 degrees C, it does indeed dissolve. So, you have to show that you at least attempted. And even then, the FDA may say, well, since you only did 2 solvents, we want you to go do something like 10% ethanol in water, So it's harder to do that. Yeah, it's a good question for sure. Yeah, I wish we could just all put our scientist hat on and make good, sound scientific arguments and have that just work. But I think, the FDA is a little bit spooked right now about it. Yeah.
What kind of criteria are we looking for to leverage the extractable leachable testing from one of our devices to a next generation device?
Yeah. So what if you have a device with extractables and leachables data and you want to leverage that on like the next generation of a device or whatever's down the road? Things can get tricky because like if that extractables data is going to be used to mitigate a specific risk like cancer, then it's very likely that the extractables testing that you did in the past doesn't meet the FDA's criteria today. And so what you could do is you could do a comparative study where you do new extractables to the modern standard side by side and compare them, but that's really burdensome and would take a long time. It would almost be worth it just to test the new device again if that was the case. That is kind of like a generic answer. Certainly it depends on the details for sure, but that's the problem that we're running into right now is we'll have devices that were run in 2018. The sensitivity was down to the TTC only. We know the FDA wants the AET, and so we can't really leverage that because there could be something between the AET and the TTC that we didn't measure before and we would miss. So, yeah. Okay, thanks.